Human evolutionary genetics studies how one human genome differs from another human genome, the evolutionary past that gave rise to it, and its current effects. Differences between genomes have anthropological, medical and forensic implications and applications. Genetic data can provide important insight into human evolution. Origin of apes. Biologists classify humans, along with only a few other species, as great apes. The living hominidae include 2 distinct species of chimpanzee, 2 species of gorilla, and 2 species of orangutan. The great apes with the family Hylobatidae of gibbons form the superfamily Hominoidea of apes. Apes, in turn, belong to the primate order, along with the Old World monkeys, the New World monkeys, and others. Data from both mitochondrial DNA and nuclear DNA indicate that primates belong to the group of eukaryotic glias, together with Rodentia, Lagomorpha, Ermaptera, and Scandentia. This is further supported by alu-like short interspersed nuclear elements which have been found only in members of the eukaryotic glias. Cladistics. A phylogenetic tree is usually derived from DNA or protein sequences from populations. Often, mitochondrial DNA or Y chromosome sequences are used to study ancient human demographics. These single locus sources of DNA do not recombine and are almost always inherited from a single parent, with only one known exception in mtDNA. Individuals from closer geographic regions generally tend to be more similar than individuals from regions farther away. Distance on a phylogenetic tree can be used approximately to indicate genetic distance. The genetic difference between humans and chimps is less than 2%, or 20 times larger than the variation among modern humans. Temporal remoteness of the most recent common ancestor. The mitochondrial most recent common ancestor of modern humans lived roughly 200,000 years ago. Latest common ancestors of humans and chimps between 4 and 7 million years ago. Speciation of humans and the African apes. The separation of humans from the closest relatives, the non-human apes, has been studied extensively for more than a century. 5 major questions have been addressed. Which apes are our closest ancestors? When did the separations occur? What was the effective population size of the common ancestor before the split? Are there traces of population structure preceding the speciation, a partial admixture succeeding it? What were the specific events prior to and subsequent to the separation? General observations. As discussed before, different parts of the genome show different sequence divergence between different hominoids. It has also been shown that the sequence divergence between DNA from humans and chimpanzees varies greatly. For example, the sequence divergence varies between 0% to 2.66% between non-coding, non-repetitive genomic regions of humans and chimpanzees. Additionally, gene trees, generated by comparative analysis of DNA segments, do not always fit the species tree. Summing up, the sequence divergence varies significantly between humans, chimpanzees and gorillas. For most DNA sequences, humans and chimpanzees appear to be most closely related, but some point to a human-gorilla or chimpanzee-gorilla clade. The human genome has been sequenced, as well as the chimpanzee genome. Humans have 23 pairs of chromosomes, while chimpanzees, gorillas, and orangutans have 24. Human chromosome 2 is a fusion of 2 chromosomes 2A and 2B that remained separate in the other primates. Divergence times. The divergence time of humans from other apes is of great interest. One of the first molecular studies, published in 1967, measured immunological distances between different primates. Basically, the study measured the strength of immunological response that an antigen from one species induces in the immune system of another species. closely related species should have similar antigens and therefore weaker immunological response to each other's antigens. The immunological response of a species to its own antigens was set to be 1. The ID between humans and gorillas was determined to be 1.09. That between humans and chimpanzees was determined as 1.14. However, the distance to 6 different Old World monkeys was on average 2.46, indicating that the African apes are more closely related to humans than to monkeys. The authors consider the divergence time between Old World monkeys and hominoids to be 30 million years ago, based on fossil data, and the immunological distance was considered to grow at a constant rate. They concluded that divergence time of humans and the African apes to be roughly 5 Maya. That was a surprising result. Most scientists at the time thought that humans and great apes diverged much earlier. The gorilla was, in ID terms, closer to human than to chimpanzees. However, the difference was so slight that the trichotomy could not be resolved with certainty. Later studies based on molecular genetics were able to resolve the trichotomy. Chimpanzees are phylogenetically closer to humans than to gorillas. However, some divergence times estimated later do not substantially differ from the very first estimate in 1967, but a recent paper puts it at 1114 Maya. Divergence times and ancestral effective population size. Current methods to determine divergence times use DNA sequence alignments and molecular clocks. Usually the molecular clock is calibrated assuming that the orangutan split from the African apes 1216 Maya. Some studies also include some Old World monkeys and set the divergence time of them from hominoids to 2530 Maya. Both calibration points are based on very little fossil data and have been criticized. If these dates are revised, the divergence times estimated from molecular data will change as well. However, the relative divergence times are unlikely to change. Even if we can't tell absolute divergence times exactly, we can be pretty sure that the divergence time between chimpanzees and humans is about 6-fold shorter than between chimpanzees and monkeys. One study used 15 DNA sequences from different regions of the genome from human and chimpanzee and 7 DNA sequences from human, chimpanzee and gorilla. They determined that chimpanzees are more closely related to humans than gorillas. Using various statistical methods, they estimated the divergence time human-chimp to be 4.7 Maya and the divergence time between gorillas and humans to be 7.2 Maya. Additionally, they estimated the effective population size of the common ancestor of humans and chimpanzees to be 100,000. This was somewhat surprising since the present-day effective population size of humans is estimated to be only 10,000. If true, that means that the human lineage would have experienced an immense decrease of its effective population size in its evolution. Another study sequenced 53 non-repetitive intergenic DNA segments from a human, a chimpanzee, a gorilla, and orangutan. When the DNA sequences were concatenated to a single long sequence, the generated neighbor joining tree supported the Homo panclade with 100% bootstrap. When three species are fairly closely related to each other, the trees obtained from DNA sequence data may not be congruent with the tree that represents the speciation. The shorter internodal time span, the more common are incongruent gene trees. The effective population size of the internodal population determines how long genetic lineages are preserved in the population. A higher effective population size causes more incongruent gene trees. Therefore, if the internodal time span is known, the ancestral effective population size of the common ancestor of humans and chimpanzees can be calculated. When each segment was analyzed individually, 31 supported the Homo panclade, 10 supported the Homo gorillaclade, and 12 supported the pan-gorillaclade. Using the molecular clock, the authors estimated that gorillas split up first 6.2 to 8.4 ma and chimpanzees and humans split up 1.6 minus 2.2 million years later, 4.6 to 6.2 ma. The internodal time span is useful to estimate the ancestral effective population size of the common ancestor of humans and chimpanzees. A parsimonious analysis revealed that 24 loci supported the Homo panclade, 7 supported the Homo gorillaclade, 2 supported the pan-gorillaclade, and 20 gave no resolution. Additionally, they took 35 protein coding loci from databases. Of these, 12 supported the Homo panclade, 3 the Homo gorillaclade, 4 the pan-gorillaclade, and 16 gave no resolution. Therefore, only 70% of the 52 loci that gave a resolution support the correct species tree. For the fraction of loci which did not support the species tree and the internodal time span they estimated previously, the effective population of the common ancestor of humans and chimpanzees was estimated to be 52,000 to 96,000. This value is not as high as that for the first study, but still much higher than present-day effective population size of humans. A third study used the same data set that Chen and Li used but estimated the ancestral effective population of only 12,000 to 21,000, using a different statistical method. Genetic differences between humans and other great apes. The alignable sequences within genomes of humans and chimpanzees differ by about 35 million single nucleotide substitutions. Additionally, about 3% of the complete genomes differ by deletions, insertions, and applications. Since mutation rate is relatively constant, roughly 1/2 of these changes occurred in the human lineage. Only a very tiny fraction of those fixed differences gave rise to the different phenotypes of humans and chimpanzees, and finding those is a great challenge. The vast majority of the differences are neutral and do not affect the phenotype. Molecular evolution may act in different ways, through protein evolution, gene loss, differential gene regulation, and RNA evolution. All are thought to have played some part in human evolution. Gene loss. Many different mutations can inactivate a gene, but few will change its function in a specific way. Inactivation mutations will therefore be readily available for selection to act on. Gene loss could thus be a common mechanism of evolutionary adaptation. 80 genes were lost in the human lineage after separation from the last common ancestor with the chimpanzee. 36 of those were for olfactory receptors. Genes involved in chemoreception and immune response are overrepresented. Another study estimated that 86 genes had been lost. A gene for type 1 hair keratin was lost in the human lineage. Keratins are a major component of hairs. Humans still have 9 functional type 1 hair keratin genes, but the loss of the particular gene may have caused the thinning of human body hair. The gene loss occurred relatively recently in human evolution, less than 240,000 years ago. Stedman et al. Stated that the loss of the sarcomeric myosin gene MYH16 in the human lineage led to smaller masticatory muscles. They estimated that the mutation that led to the inactivation occurred 2.4 million years ago, predating the appearance of Homo ergaster erectus in Africa. The period that followed was marked by a strong increase in cranial capacity, promoting speculation found that the loss of the gene may have removed an evolutionary constraint on brain size in the genus Homo. Another estimate for the loss of the MYH16 gene is 5.3 million years ago, long before Homo appeared. Other CASPASE12 have cysteine aspartate proteinase. The loss of this gene is speculated to have reduced the lethality of bacterial infection in humans. Gene addition. Segmental duplications have had roles in creating new primate genes and shaping human genetic variation. Human-specific DNA insertions. When the human genome was compared to the genomes of 5 comparison primate species, including the chimpanzee, gorilla, orangutan, gibbon, and macaque, it was found that there are approximately 20,000 human-specific insertions believed to be regulatory, while most insertions appear to be fitness-neutral. A small amount have been identified in positively selected genes showing associations to neural phenotypes and some relating to dental and sensory, perception-related phenotypes. These findings hint at the seemingly important role of human-specific insertions in the recent evolution of humans. Selection pressures. Human-accelerated regions are areas of the genome that differ between humans and chimpanzees to a greater extent than can be explained by genetic drift over the time since the two species shared a common ancestor. These regions show signs of being subject to natural selection, leading to the evolution of distinctly human traits. Two examples are HAR1F, which is believed to be related to brain development, and HAR2 that may have played a role in the development of the opposable thumb. It has also been hypothesized that much of the difference between humans and chimpanzees is attributable to the regulation of gene expression rather than differences in the genes themselves. Analyses of conserved non-coding sequences, which often contain functional and thus positively selected regulatory regions, address this possibility. Sequence divergence between humans and apes. When the draft sequence of the common chimpanzee genome was published in the summer 2005, 2,400 million bases were sequenced and assembled well enough to be compared to the human genome. 1.23% of this sequence differed by single base substitutions. Of this, 1.06% or less was thought to represent fixed differences between the species, with the rest being variant sites in humans or chimpanzees. Another type of difference, called indels, accounted for many fewer differences, but contributed to 1.5% of unique sequence to each genome. Since each insertion or deletion can involve anywhere from one base to millions of bases, a companion paper examined segmental duplications in the two genomes, whose insertion and deletion into the genome account for much of the indel sequence. They found that a total of 2.7% of euchromatic sequence had been differentially duplicated in one or the other lineage. The sequence divergence has generally the following pattern: human chimp and LT, human gorilla and LT, human orangutan, highlighting the close kinship between humans and the African apes. ALU elements diverge quickly due to the high frequency of CpGD nucleotides, which mutate roughly 10 times more often than the average nucleotide in the genome. The mutation rate is higher in the male germline. Therefore, the divergence in the Y chromosome, which is inherited solely from the father, is higher than in autosomes. The X chromosome is inherited twice as often through the female germline as through the male germline, and therefore shows slightly lower sequence divergence. The sequence divergence of the XQ13.3 region is surprisingly low between humans and chimpanzees. Mutations altering the amino acid sequence of proteins are the least common. In fact, till 29% of all orthologous proteins are identical between human and chimpanzee. The typical protein differs by only 2 amino acids. The measures of sequence divergence shown in the table only take the substitutional differences, for example from an A to a G, into account. DNA sequences may however also differ by insertions and deletions of bases. These are usually stripped from the alignments before the calculation of sequence divergence is performed. Genetic differences between modern humans and Neanderthals. An international group of scientists completed a draft sequence of the Neanderthal genome in May 2010. The results indicate some breeding between modern humans and Neanderthals, as the genomes of non-African humans have 1 to 4% more in common with Neanderthals than do the genomes of sub-Saharan Africans. Neanderthals and most modern humans share a lactose-intolerant variant of the lactase gene that encodes an enzyme that is unable to break down lactose in milk after weaning. Modern humans and Neanderthals also share the FOXP2 gene variant associated with brain development and with speech in modern humans, indicating that Neanderthals may have been able to speak. Chimps have 2 amino acid differences in FOXP2 compared with human and Neanderthal FOXP2. Modern humans. Molecular biologists starting with Wesley Brown on mtDNA and Alan Wilson on mtDNA have produced observations relevant to human evolution. Age of the common ancestor by estimating the rate at which mutations occur in mtDNA. The age of the common ancestral mtDNA type can be estimated. The common ancestral mtDNA links mtDNA types that have diverged by an average of nearly 0.57%, assuming a rate of 2% to 4% per million years. This implies that the common ancestor of all surviving mtDNA types existed 140,000–290,000 years ago. Most, but not all, scientists in the relevant fields consider this observation robust. This common direct ancestor in the line of mother to daughter of all extant humans has become known as Mitochondrial Eve. The observation that the mTMRC is the direct matrilineal ancestor of all living humans does not mean either that she was the first anatomically modern woman, nor that no other women lived concurrently with her, nor even excluding the existence of other women being ancestors of today's people. Other women would have lived at the same time and passed nuclear genes down to living humans, but the mitochondrial lineages were lost over time. This could be due to events such as producing only male children. African origin for modern humans. There is evidence that modern human mtDNA has an African origin. We infer from the tree of minimum length that Africa is a likely source of the human mitochondrial gene pool. This inference comes from the observation that one of the 2 primary branches leads exclusively to African mtDNAs, while the second primary branch also leads to African mtDNAs. By postulating that the common ancestral mtDNA was African, we minimize the number of intercontinental migrations needed to account for the geographic distribution of mtDNA types. The broad study of African genetic diversity headed by Sarah Tishkoff found the San people to express the greatest genetic diversity among the 113 distinct populations sampled, making them one of 14 ancestral population clusters. The research also located the origin of modern human migration in southwestern Africa, near the coastal border of Namibia and Angola. Y chromosome lineages. The Y chromosome is much larger than mtDNA, and is relatively homogeneous, therefore it has taken much longer to find distinct lineages and to analyze them. Conversely, because the Y chromosome is so large by comparison, it holds more genetic information. Y chromosome studies show similar findings to those made with mtDNA. The estimate for the age of the ancestral Y chromosome for all extant Y chromosomes is given at about 70,000 years ago and is also placed in Africa. The individual who contributed this Y chromosomal heritage is sometimes referred to as Y chromosome Adam. The difference in dates between Y chromosome Adam and mitochondrial Eve is usually attributed to a higher extinction rate for Y chromosomes due to greater differential reproductive success between individual men, which means that a small number of very successful men may produce many children, while a larger number of less successful men will produce far fewer children. At a recent conference at the American Society of Human Genetics, there was a study presented by Melissa Wilson Sayers from the University of California, Berkeley, which called into question the amount to which people had more female ancestors than male, while it didn't deny that men have historically produced at a higher rate than females. A polygamous mating system doesn't explain how the Y chromosome is so uniform. Other people have suggested it's as few as 4 females for 1 male. We find that it's probably skewed, but it's more like 4 females to 3 males, said Sayers. Ancestral components. Modern DNA sequencing has identified various ancestral components in contemporary populations. A number of these genetic elements have West Eurasian origins. They include the following ancestral components with their geographical hubs and main associated populations.